Oh, is that too dark? There is only one. Oh, the far. And then do what? Okay. Well, thanks for the introduction, Megan. This is actually the first time I've ever given a talk in the physics department, ever. And as you can see, I don't know how the lights work. I have given a talk in the biology department and the chemistry department, but that was back in 1999, I guess. So I guess it's about time. Last week, or at the weekend, I was at a photosynthesis conference up in Indiana, and there was a bunch of graduate students there who were avid home brewers, and they made these concoctions that were essentially irresistible to a Scotsman. And I'm still suffering a little from that, so my brain is a little cloudy. Anyway, if a cloudy brain leads to a cloudy presentation, please feel free to just shout out questions and interrupt, no problem. I prefer you ask questions as we're just moving along. Okay, so let me, just as a start, introduce you to this very well-known statistic. More energy from the sun hits the Earth in 1 hour than is used by mankind in 1 year. Hardly any of that solar energy is utilized. To really utilize solar energy, you first have to understand it. And plants know how to utilize solar energy very well, and it's relatively it's quite very widely believed that understanding how things happen in plants will lead to greater solar energy utilization in the future. At least that's the idea. So that's one motivation for maybe studying solar energy conversion in native systems. So in my lab, there's several projects ongoing. In recent years, I've been doing a lot of FTIR microscopy and imaging on biological cells. One of these projects is to study harmful algal blooms and try and assess whether infrared spectroscopy can be useful for monitoring the progression of blooms. And the reason I got interested in this is the park right next to my house, Lake Claramere, has been known to have fish kills because of algal blooms in the past. And this is measurements that we made a couple of years ago, and you can see this green slime here. And the idea is, can we say things about algal blooms based on the IR spectra of the material that's there? Now that really means building huge libraries of spectra for many types of algae, and that's where we are with that. Another project we have going is to actually look at lipid production in microalgal cells. These lipids are precursors for biodiesel, and so one of the ideas is to take a look at stuff like that. And we have been working with a guy in Florida, Reza Razagifard, on looking at this strain Neochloris minuta. When you grow these cells in the absence of nitrogen, it hugely overproduces lipids. And so we're actually interested in using infrared spectroscopy as a probe of the lipid contents in biological cells. And so that's another thing that we have ongoing. Oops. And so a lot of the work we're doing nowadays is in time resolved visible and infrared spectroscopy to probe the solar conversion reactions in photosynthesis. We measure IR spectra, and then we are also involved in doing a lot of QM/MM calculations to simulate the experimental spectra. I probably won't talk about any of that today, and I'll focus mainly on the time resolved infrared spectroscopy. So anyway, to start, this gives you a broad overview of the photosynthetic process. Carbon dioxide and water are absorbed by a plant, and light leads to the conversion of these inputs into sugar. Sugar is essentially the biofuel. It's a solar fuel. So when you think about solar conversion, you sometimes think about solar panels and the production of electricity for me, I tend to think more about the production of fuels, as in like, so carbohydrates in a natural system. And oxygen is a byproduct in photosynthesis, a waste product, if you like, which is, that's utilized by us. So this is the overall process, and it's quite misleading. There's underlying this process, there's about 100 other processes. And so it's actually a very, very complicated set of processes, which is just shown in a very simplified form here. So the question is, where does photosynthesis take place? It turns out that the oxygenic photosynthesis that occurs in a plant is almost essentially identical to that in cyanobacteria. And this shows a picture of a cyanobacterium. This is a unicellular cyanobacterium. And it turns out that they're easy to grow and manipulate, so it's a lot better to work on cyanobacteria than it is to work on plants. Let's see, which way am I going? Oh. So we actually grow these cyanobacterial cells in our lab. We have wild-type strains, Synicocystis 6803, and we also work with mutant strains that allow us to modify the photosynthetic machinery. So in a cyanobacterial cell, around the periphery of the cell, you see these lipid bilayers, these stacked bilayers. They're called thylakoid membranes. And it's in these thylakoid membranes that the photosynthetic apparatus resides. And so this shows a picture of one of the thylakoid membranes, and embedded in this membrane are these large protein complexes called photosystem 1 and photosystem 2. And this is where the initial light energy is absorbed. The two photosystems work independently, but they cooperate in splitting water, and the electrons from water end up with carbon dioxide in glucose. And so following light excitation, what really happens, the basic mechanism is very well known. It's similar in all systems. An electron is transferred across the membrane, so you've generated this electrochemical gradient, and there's energy in that gradient. This is just a capacitor, right? And so different enzymes can access that energy to produce different products. One of the products would be ATP. And so that's the actual basic mechanism of solar energy conversion in plants. Photosystem 2 is the site of oxygen evolution. It's where water is split. Photosystem 1, it generates reducing products that are used to reduce carbon dioxide, add protons to carbon dioxide, and turn it into the carbohydrate glucose. So we'll be mainly interested in photosystem 1. And this shows, how do I make the movie start? Oh, there we go. If I do that. Okay, don't know how to make the movie start. So anyway, this is a picture of the photosystem 1, and these proteins span the membrane. And embedded in this protein complex, there's a bunch of electron transfer cofactors, which would open up if the movie worked, but that's fine. This shows a picture of the electron transfer cofactors that are embedded in a protein. So the protein binds different pigments in place. And so in photosystem 1, light comes in, and it causes an electron to be transferred from P700 hopping via these pigments across the membrane. In photosystem 1, it's only become shown in the last few years that electrons can actually go both ways. So if you can actually control which way an electron goes, you have the basis of a sort of nanomolecular optoelectronic device. If you can actually figure out how can you control how much electrons go this way compared to that way? And that was one of the interesting things about this discovery, which happened in the last 10 years or so. So anyway, P700 is a pair of chlorophyll molecules. The electron hops, and the secondary electron acceptor called A1 is a quinone. These FX, Fa, and Fb are iron sulfur clusters. Following excitation of P700, the electron is over there within 300 nanoseconds. And I say the efficiency is 98%. What I mean is, once the electrons get over there, there's terminal acceptors that will take the electron out. The electron is not coming back the way. And so that's what I really mean by efficiency. The electron is not coming back the way. And that's what limits a lot of artificial devices, is this recombination reaction. So in a native photosystem, the electron will just go way over here, and it will never return to its starting position. And so the energy will be captured. So the question is, why do electrons? They move very rapidly across the membrane, and they do it with like very high efficiency. The question is why? And of course, it's related to the properties of the pigments, and these properties are bestowed on the pigments by the protein interactions. And so we have and have had various projects to study the different pigments that are involved in electron transfer. This one's not active, but we have some work going on the primary electron donor, and we also have work. Most of the work these days is on the A1 pigment that occupies the so-called A1 binding site. So the electron gets to the pigment, and it's moved off very quickly. We want to actually capture the electron as it's on the pigment, and so we have to use time-resolved spectroscopy. And we use both time-resolved infrared and visible absorption spectroscopy. And that's what I'll talk about here. In photosystem 1, the native system, the native pigment, this A1 pigment is called a phylloquinone molecule. It's a 2-methyl-3-5-1,4-naphthoquinone. Turns out it's the most reducing quinone in biology. And the question is just how reducing. It's so reducing it's almost impossible to measure using electrochemical techniques. But from the bioenergetics, we will be able to assess what the potential of that pigment is. Photosystem 1 has to be highly reducing because it has to reduce carbon dioxide to glucose. And this is essentially one of the most reducing quinones in biology. Okay. So one way to, in our time-resolved measurement, at least for visible spectroscopy, we're going to use a pulse laser to excite the sample, and we're just going to use a light source and use filters to probe at different wavelengths. Different probing wavelengths different wavelengths will probe different species. So if we probe at, say, 487 nanometers, we'll actually be probing the electron as it's on the A1 pigment, the phylloquinone molecule. If we excite at, say, if we probe at 703 nanometers, we will be probing the P700 cation radical, and as that radical decays. And so these two different wavelengths. So here you can see this would be at 487. As you flash the sample, you get a positive absorption change, and that's due to the electron being on the pigment. And that absorption change recovers as the electron leaves the pigment. And so that's kind of what these traces will mean. So if you want to probe electron transfer processes, you need some way to manipulate them and look at how they change under various types of manipulation. And we want to modify the pigments in the binding site. So one way to do that is we have a mutant. So phylloquinone is synthesized via enzymes in the protein, in the cell. And so if we can disrupt the genes that code for the synthesis of these proteins, we can stop the synthesis of phylloquinone. If we do that, we expect the binding site to be empty. But it turns out when we make that mutant, it's called a MEN-B mutant. When we make that mutant, a plastoquinone molecule is incorporated into the binding site. I'll show you a structure of plastoquinone later. So that plastoquinone is not very well bound in the binding site, and it's easy to displace it. And we can simply by incubating in the presence of a bunch of other quinones, we can incorporate different quinones into the binding site. These different quinones have different potentials, and so we can, essentially what we will be doing is modifying the energy levels by putting different quinones in, and then probing the energetics with these different energy levels. So what I'm showing over here, it's written in terms of midpoint potential, but you can think, if you're a physicist, you can think about it just in terms of, the energy of the electron, if you like. if you go from here to here, that's about 1.85 electron volts, that corresponds to a 700 nanometer photon. P700 absorbs at 700 nanometers, and a 700 nanometer photon causes that change. And so this state represents the electron, the first electron transfer, the electron transfer to the quinone, and further electron transfer steps. And these are rough estimates of the actual energy levels involved. So you can see if I put in these different quinones, I'm going to play around with that energy level, and so I can then look at the energetics when I do such a thing. So it's essentially moving up and down like that. And so for today, I put in these different quinones. Oh, and a shout out, actually, Maged Hanari in the chemistry department synthesized that quinone for us. So a lot of this work involves people synthesizing quinones as well that don't exist. Well, that's not a very uncommon quinone, but anyway. So roughly what we expect is we're going to change the energy levels over here. So you can see forward electron transfer from here to here is now uphill, and so that's going to change the time constants associated with electron transfer. And okay, so at room temperature, let me show you some experimental data. At room temperature, if we put quinones in that tend to lead to a raising of this energy level, so these types of quinones, what we see when we do a measurement at room temperature, at 487 nanometers, we see a nanosecond decay and a microsecond decay. These correspond to electron transfer down the two different branches in photosystem 1. So that's at 487 where we're probing the electron on the quinone molecule. On a longer time scale, on tens of milliseconds, we see the recovery of P700 plus. So we're seeing two different things, which is telling us about what the energetics have to be. And so this is what's actually happening. We're getting forward electron transfer to this terminal cofactor, and then recombination. You can't actually have recombination from FB back to the ground state. The distance is just too far. It would take weeks for that to actually happen. So what actually happens is the A1 minus radical is repopulated, and then you have recombination back to the ground state from there. And we know that has to be the case because this recombination changes when we change the pigment that's in the binding site. And so that's what we see at room temperature for this class of quinones. If we look at, what did I do? If we look at other quinones with where the energy level is much lowered, we call these high potential quinones because they're less negative than the other ones. And in that case, what we find is if you look at the A1 minus radical, it actually decays with the same time constant as P700 recovers. So that tells you that you're looking at a direct recombination reaction between A1 minus and P plus. And so that's for the high potential quinones, or the less negative quinones, if you like. And so in that case, what we're actually looking at, because this energy level is so low, we can't have electron transfer uphill here, and so we just see a recombination reaction. So we can distinguish what's going on by looking at these different recombination reactions. So if you look at, so we have two different kinds of data. So at 298K, what we see is, so as you make the energy level more and more negative, what you find is the time constant for the forward electron transfer process gets shorter and shorter and shorter. That means the rate goes up and up and up. So that's what you expect. The difference in energies between the two states, you're making that energy difference larger. That's what, in chemical terms, you would say the driving force or the free energy is larger. And when the free energy is larger, the rate is faster. It's like you're going more downhill, you get a faster rate. And so that's kind of what we see there. So as the driving force goes up, the rate goes up for the forward electron transfer process. Now, if we look at the recombination reactions, the opposite is happening. The quinones with a very negative energy level, the rate is very long. and it becomes shorter and shorter and shorter as the energy gap decreases, which is very counterintuitive. That's just not what you would normally expect. Now, to study, if you really want to study electron transfer well, you have to be able to do measurements at different temperatures. And so we've also done measurements at 77 K for all of these different quinones. And it turns out in photosystem 1 at 77 K, for the most part, forward electron transfer is blocked, and you're looking only at the recombination reaction. And it turns out we've done other measurements where, and it's generally this recombination reaction occurs only on the A branch, not on the B branch. So this is really nice. You just have a single electron transfer process between two species, so the rate that you observe will be directly related to the rate of electron transfer. And so that's pretty nice. So this shows data for all of these different quinones probing at 700 nanometers, because we're looking only at a recombination reaction. And you can see, so the native system with a phyloquinone molecule in the binding site is the black line here, and that corresponds to that recombination. That's about 350 microseconds. If we look at an anthraquinone molecule, that's really pushing this energy level up the way, so you can see the downhill, the energy difference between the two states is much larger, but the rate has become much, much slower. The time constant has become much longer. It's about 800. That should be wherever you see a milli, change it to a micro. That should be a microsecond. So that's 800 microseconds. And when you have a very small energy gap between here and here, corresponds to something like a dichloronaphthaquinone in the binding site, and that rate is about 70 microseconds. So you can see, as you make the energy gap smaller and smaller and smaller, the time constant gets shorter and shorter and shorter, which means the electron transfer rate has gone up. So let's see what we have. So at 77 K, the driving force increases, but the electron transfer rate has decreased. That's like totally non-intuitive. It's like going to a ski slope, seeing the slope get steeper, but you go down it slower anyway. And so it's quite non-intuitive. And so we want to address this question now. So in a real system, you want to have the forward electron transfer. You want to get this electron across the membrane. So you want this to happen, but there's hardly any driving force. The recombination reaction has a much larger driving force. But this back reaction doesn't occur. And so the question, even though it's way downhill, so the question is, why does that happen? So a roadmap from where I'm going from here is, so I'll tell you something about electron transfer theory, and we want to simulate all of our data, and we have a lot of data now. We have forward electron transfer rates, recombination rates at room temperature for 10 different quinones incorporated, and also at 77 K as well. We want to try and simulate all of that within one single model. And so that model should allow us to estimate the quinone midpoint potentials, and also then we can start talking about the reaction-free energies and what mechanisms are appropriate. So that's where I'm going now. So an electron transfer process, you have a donor and acceptor, and of course the electron goes from the donor to the acceptor. That's an electron transfer process. And in the 1950s, Rudy Marcus developed that theory of electron transfer. And in 1992, he received the Nobel Prize primarily because of experimental verification of this theory that he came up with. Ooh. Okay, well that's not good. So basically, you have a donor and acceptor, and there's an electronic coupling between these pigments, and that the rate of electron transfer can be calculated using Fermi's golden rule, which leads to an equation which is, there's an equation there. Okay, well, fortunately, Moser-Dutton came out Les Dutton and Chris Moser in the 90s came out with a great simplification of this much more complicated equation. So what they did is there's a bunch of parameters in here. Maybe you can still see them. There's a beta parameter which talks about the medium between the donor and acceptor. R is the distance between the pigments. Lambda, delta G is a free energy. Lambda is a reorganization energy. And what that is if you have a radical pair state, plus charge, minus charge, the environment is going to be modified in the presence of these charges, and the amount of energy required to make that modification is called a reorganization energy. And what Moser Dutton did is they came up with this much, much simpler formalism. So this is K script ET, so it's the rate of electron transfer. The log of the rate is equal to this. This 15 comes from parameterizing a whole bunch of these quantities in the top equation that they got from types of measurements. And R is the edge-to-edge distance between the pigments. Delta G and lambda are given there. Delta G plus lambda, that's actually squared. So the superscripts are not printing out very nicely here. Anyway, it turns out that if you look at that equation, so this is delta. This is really the log of the rate as a function of delta G, the free energy, is just an inverted parabola. Yep. That's yeah. So from various measurements, they measured beta, and from beta you can get you can extrapolate a curve back to 0, and you can get V naught and beta out of that. It's in there as a parameter with some value, but if you want to change that value, you can't really use this equation. No, they did it. There's lots and lots of different experiments on different kinds of electron transfer processes, and they were able to estimate beta and V naught. And it's all encompassed factor 15. But if you want to, you don't want to really use that factor 15 if you want to, estimate these parameters. So you can see so this is called the Marcus curve, where you plot the log of the rate. The energy goes up as the distance between the two energy levels. The electron transfer rate goes up. That's called the normal region. And when minus delta G equals lambda, this term equals 0, and you get a maximum possible electron transfer rate. However, if you continue to increase delta G, if you make delta G the difference between the energy levels bigger and bigger and bigger, the rate starts to go down again. And this is called the inverting region. And this is what Marcus, and this is what was shown experimentally in the 80s by Kloss and Miller and their co-workers. And I believe that's what led to Marcus getting the Nobel Prize for this work. So in his Nobel lecture, Marcus actually said that this inverted region mechanism could be important for promoting very efficient electron transfer. Because here's what's happening. You want to get electron transfer between a donor and an acceptor that are very close in energy. But that's the productive pathway. That's the way you want to go. The unproductive pathway back to the ground state, you want to inhibit that process if you can. So he said, yeah, this could be useful in photosynthesis for generating very high efficiency solar energy capture. It turns out that inverted region electron transfer is never actually in any native photosynthetic system. You can generate various kinds of modified systems, but what's really important is the native system, right? What happens in the real world. And it's never actually been demonstrated in a native system. So the question is, for at least the work that we were doing, does inverted region electron transfer, does it contribute to photosynthetic efficiency and the actual quantum yield? And what's primarily of importance is it working in the native system at room temperature? And the problem with photosystem 1 is because there's so many reactions going down two branches, what the actual intrinsic rate of electron transfer is very complicated because you have multiple processes, and what you see in an experiment is some convolution of all these different processes. So you need a kinetic model to to where you can input the time constants and then you can estimate them via solving some sets of differential equations. And that's what we did. So this is our kinetic model where we've included all the forward and reverse rates. We've included electron transfer on both branches. It's bidirectional. And thermodynamic equilibrium is pretty much included in this model. And we solved a more complex, so the Moser-Dutton equation, or the Marcus equation it came from, is really a classical expression. So this is semi-classical, where we've incorporated the fact that different normal modes, vibrational frequencies, can be coupled to the electron transfer process. Rho is a packing density, which can be calculated based on the atoms between the donor and acceptor. If you have crystal structures, you can estimate that. I should point out so all the pictures I've showed you of the electron transfer cofactors, they all come from crystal structures. So we know the distances between the pigments, we know what the intervening medium looks like, and so we can estimate a whole bunch of parameters, like rho and r. And to model backward electron transfer, we just say the free energy is minus the forward free energy. And so that's fine. But the important point is that the intrinsic rate constant, the rate of electron transfer, in no way needs to correspond to the observed rate. And nearly every paper we publish, all of the reviewers' criticisms essentially follow from the fact that the confusion that the observed rate and the intrinsic rate are the same thing. And in many systems they are, but in photosystem 1 at room temperature they're not. And so that needs to be accounted for. And so here's our model at 298 K. The electrons have hit the quinone in 50 picoseconds. And at room temperature, we know the distribution of A1A and A1B is about 50-50. If we make it 80-20, it doesn't actually make any difference in the rate equations that we solve. And at low temperature, we've done a whole bunch of experiments in our lab where we've shown that electrons go down, or recombination occurs down the A branch in about 95% of the samples. So these are the kinetic models that we're going to work with. And this is just, picture of coupled differential equations. We're just going to solve them. It's pretty straightforward. And what we're going to plot is the population evolution of the actual radical pair states, because that's what you get from the experiment. And so you need to solve these equations, and then you can work back and find out what the intrinsic rate constants actually are. And so that's the equation we solved. We know temperature. We know that we estimate what the mode coupled to electron transfer is. That's h bar omega. We know the distance between the pigments. Well, that's r. I don't know what that's supposed to be in there. Whatever that is. So anyway, we can get the K's, we can get the rates of electron transfer from using this equation. We can substitute that into the differential equations. We can solve the differential equations and get that population evolution. And we can then compare that to experiment. And so here's just a kind of procedural, a picture of the procedure that we follow. So initially, the way we work it, okay, we put in the native quinone. We make sure we can simulate the forward electron transfer at room temperature. If that works, great. We can then estimate if we can get the right numbers for the recombination reactions at room temperature. And if that works pretty nicely, we then go make sure that our model works for the low temperature reactions as well. And then we go through the whole procedure again for using non-native quinones in the binding site. And at every step, we have to be able to satisfy all of these things. If we can't, then we have to go back and change some of the parameters in the model. So anyway, this is just a simulation here, and this shows the population of the total amount of phyloquinone anions, if you like. And what you can see is the population evolution. It starts in the nanosecond scale for anthraquinone in the binding site, And the rate gets longer as you go to quinones with higher and higher potentials. So that's for the forward electron transfer. For the recombination reactions, we're looking at P700 recovery, and that's shown here by the black lines. And for that, you can see a decrease in the recombination rate as you go to quinones with lower and lower potential. So that's an increase in the actual ET rate when the energy difference is smaller. So this agrees very well with the experiment. And at 77K, we can do the same thing. We can look at the recombination rate. We can see the recombination rate is going up and up and up as the energy level difference is going down and down and down. So we can explain the calculations are in pretty good agreement with the experiment. So with all of this data, we now have an estimate. We can build a Marcus curve. I forgot to mention the actual curve where I showed you the inverted region. It's called the Marcus curve, where it's a plot of the log of the electron transfer rate against the free energy. And so this is what the observed stuff looks like at 77K. And there are some modifications to when you go from intrinsic to observed, mainly for quinones with very low potential. And that's what the actual curve looks like. And if you plot that, this is assuming that the mode coupled to electron transfer has an energy of 70 millielectron volts, which is quite standard. From various types of magnetic resonance experiments, you can actually measure the distance between the pigments very accurately. And the EPR measurements are accurate to about 0.2 angstroms. So if you include this 0.2 angstroms is the error bar in the y direction, that's shown there. And we can also play around with the modes that are coupled to electron transfer. We can change them, and that's shown here by the dotted line. And if you actually look at the whole curve, this is the Marcus curve, and you can see all the quinones we looked at 77K. Everything is in the inverted region, everything, which is very nice. The native system is way into the inverted region, so it's 77K. The recombination electron transfer is way into the inverted region. So, and this is actually a first demonstration of electron transfer in a native system in the inverted region. It's been shown for sort of modified systems, but never for a native system. But that's at 77K, which isn't really very useful, right? At room temperature, 298K, we can do the same kinds of calculations. And again, for both the A and the B branch recombination, the electron transfer, the recombination process is in the inverted region. So that's quinone number 4 is the native quinone. So inverted region electron transfer happens at room temperature and at low temperature in photosystem 1. Okay, so now I've kind of told you about an isolated photosystem 1 particle. Now, in the actual cell, there's peripheral donors and acceptors that will accept electrons and donate electrons. So the real goal is to know if this system is still going to work in the native whole cell, if you like. And so we've done modeling where we've included a sink for electrons, and also we can include electron donors that will donate to P700 plus. So we've done many different kinds of calculations that are much more realistic of what happens in an intact thylakoid membrane rather than a PS1 particle. And so if you look at these more detailed kinds of models, what I'm calling photosynthetic efficiency is the lack of recombination that happens. So if P700 plus doesn't go back to the ground state, if it went back to the ground state, it would go up to here. So if P700 does not recover, that's what I am equating with photosynthetic efficiency. And in this figure, the photosynthetic efficiency is essentially 98% for our modeling of an intact system with donors and acceptors, peripheral donors and acceptors included. And that's if inverted region electron transfer is included. So if we if I now say, okay, let's make the recombination reaction, let's make the electron transfer process optimal. So we change lambda, and delta G and lambda are the same. So we make it optimal, and when we do that, what we find is this like large recovery of P700. In fact, it recovers 28% of P700 recovers. So the quantum efficiencies drop to about 72% in these native systems if the electron transfer doesn't occur in the inverted region. And so we argued that, well, this is clear evidence that this mechanism is actually important for generating very high photosynthetic efficiency. Okay, so I just said that, right? So the question is, why does this happen in PS1 and it doesn't happen in other photosynthetic systems? Well, it turned like photosystem 1 is a highly reducing enzyme. The idea of photosystem 1 is to shuttle electrons into carbon dioxide, and that will make it mix with protons to basically reduce carbon dioxide, which will then be assimilated into glucose. So there's a necessity to have a very reducing environment in photosystem 1, and that's shown here by this picture. So in purple bacteria and also in photosystem 2, the terminal acceptors are about 0 volts. There's no chemistry going to be happening with these electron acceptors. But in PS1, the terminal acceptors are still around about 500-600 millivolts. And that can be used to do a lot of chemistry, like reduce carbon dioxide into glucose. And if you look at the nature of the reaction, the recombination in purple bacteria is known to be optimal. You can change this quinone, and you find that the reaction is optimal. But in PS1, the peripheral or the more terminal electron acceptors are very close in energy to the intermediates. So to make this forward reaction happen in preference to that reaction, you have to have this reaction in the inverted region. So it's a really important mechanism. So and this might be useful in the future. If you want to generate reducing products, you might want to incorporate the mechanisms associated with inverted region electron transfer. So that's why it happens in PS1 and perhaps not in other photosystems. So to summarize, you can read that. So we can monitor the population evolution of the various radical pair states on different timescales and using infrared and visible spectroscopy. I didn't really talk about any of the infrared spectroscopy. We can use the pseudo-equilibrium model. We can estimate the redox potentials of the pigments in the binding site. And these redox potentials were in the Marcus curve. And so it is very difficult to measure these redox potentials. In fact, because we can actually use the bioenergetics to specify what the midpoint potentials will be for quinones that have never been measured using electron chemistry. And we're actually doing that with someone in the chemistry department, where we have got quinones where we don't know what the potentials are, but we've measured the bioenergetics, so we can estimate what the potentials are. So we're giving him these quinones, and he's going to try and measure the electrochemistry. So I'm saying, just throw away your electrochemical gear. Let's just measure the bioenergetics, and we're good to go. Giovanni's not agreeing with me. So anyway, unproductive electron transfer occurs in the inverted region, and this is the first demonstration of that. And it's clearly an important mechanism for gaining high photosynthetic efficiency in a system that requires the pigments to be highly reducing. And of course, so the reducing nature of the pigments in PS1, you need inverted region if you want to work in a highly reducing regime. That's the final result. And this is an old picture from a while back, from a while ago. But everything I talked about today was done by this fine gentleman. Is he here? Hiroki Mikita. And this work was initially supported by the Qatar National Research Fund, and the Department of Energy saw how nice it was and decided to pick it up after that. And with that, I'll thank you for your attention.